Thank you for inviting me here. It's lovely to see you all here. Thank you for staying this long. I know it's been a very long day. It has been a very long day for me. And it has been this is your third long day, right? Or maybe fifth for some of you. So thank you for staying and listening to me. Thank you. I know Mexico is also listening. So Arte Mais Ciencia are listening. Let's begin because I have a lot of things to say and I don't have a lot of time to say it. So first, let me tell you that I'm an artist. I have sorry, I'll hold this. I have no formal background in science at all, but I have started my artistic career, so my final project for my painting degree in Lisbon was actually my first incursion into a biology laboratory, doing a residency, and I made my first project in bioart as my final project for my degree. Bioart is maybe a field. It's a very broad field. Maybe it's a community more than a field. Everybody does their own thing, so it's really not a movement in a traditional way. And there's more and more, so I began in 1999, so about 22 years ago. But since I started until now, you have more and more artists doing work with living materials. And this is one of the broadest ways of describing what bioart can be described as, which is using the living materials, whatever they may be, to produce art, to make art. And since there are many different kinds of living materials, there's lots of different technologies to manipulate them to produce art. These are some examples. They're just nice images, but there are absolutely amazing projects behind these images. So I do encourage you to look into what has been done by many artists around the world. And I wanted to also tell you that I run two institutions in Portugal. One is in Lisbon, it's Ectopia, and the main focus of Ectopia is to connect artists to scientists and their labs. So if you want to do a residency with a scientist and you want to work on a particular subject, we will be very happy to point you in the right direction, ask a few scientists around if they are interested in working with you, and connect you to them, and you can work within their labs. And then 2 hours south from Lisbon, we have Cultivamos Cultura. It's right on the edge of the Sudoeste Alentejano Natural Park. It's more focused on ecology, and it's the biggest advantage of Cultivamos Cultura is actually that it's about space. So there's lots of space. There's the park, but also the space where we are. It's an old farm, which is in a very small village in São Luís. It allows us to just play with anything. So there's lab there, which is more of a field lab, but you can do microbiology, you can do a lot of different things. You have microscopes and things like this, but also you have a very big barn in which to play with. And so sometimes you're working in a lab and you're doing things at a very small scale, but as you want to transform that information into an art piece that needs to be in an installation, in a gallery space, you need to turn it into a more humane kind of scale. And so you need to play with size. And Cultivamos Cultura is the best place to do it. I am obviously not the only institution, or we are obviously not the only institution in the world who does this. A lot of the institutions in the world, I think most of the ones that I know, are actually on academic institutions, are part of academic institutions, and they are from artists who started working in biology and then become teachers, and as professors they started their own laboratories, their own places for artists and students to work with biology, and now you have a pretty wide and pretty interesting network of places where you can have a master's degree fully funded, like in the University of Windsor or at Buffalo University in New York. The School of Visual Arts actually makes absolutely amazing summer schools. If you want to go a little bit further and do a PhD, the Ressenler Polytechnic Institute is one of the best places to be. Concordia University has a huge apparatus with the milieu where you can do biology, but also everything else that you can think of. And the institution, or the academic institution that I know that started, that was the first to create a laboratory specifically for artists, was with Dr. Jonat Zoer and Oren Katz in Western Australia in Perth. I think most of the professors that you see around this slide have at some time or another done a residency at Symbiotica in Perth. And it's the second time that we mentioned Symbiotica today, I think. There's also Arte mais Ciência at UNAM. They have an amazing group composed of designers, of scientists, of philosophers. It's, I think, one of the, I'm sure it was the first group that was set up like this. It's in a humanities department, but it connects to the Faculty of Fine Arts, and it connects to the Department of Biology in the UNAM. And of course, you have institutions which are not academic institutions that exist to produce, to exhibit, to show works like this in many places around the world. So, this is what I do. And I think it's important to tell you a few things about how I go about doing my work. So my first project was Nature with a question mark. Like I said, I was a very young art student, and it was the first time that I was in a biology lab doing a residency. It was not the first time that I was in a biology lab, but it was the first time I was doing a residency. And I was very fortunate to be encouraged by friends who were doing PhDs in biology at that time to actually ask to go and do the residency in a lab. It was not very common. I was not sure how to ask as an artist to do a residency in a science or a biology laboratory. And I think that just the same as I didn't know very well what I was doing, the scientists were not very sure what to expect from an artist in the lab. So once they found out that I was not that crazy, or that I was not going to boycott any of their experiments, they just took me as another researcher. And this was absolutely fundamental for the way that I work within a lab. I never felt like an interloper. I never felt like an appendix. I never felt like they were doing me a favor for allowing me to be there, and they never made me feel that way either. They also never made me feel like I had to make pretty images from the science that they were producing. And this was very, very important. So I had to do all the chores that all the other researchers there were doing. I had to present lab meetings just like everybody else. And I had to face questions and try to answer them to the best of my abilities. What were my intentions? Why was I choosing to do this way and not another way? What gave me the right to manipulate a living organism for art was one of the first questions that they gave me. And I was 22 at that time. So this was incredibly forming for me. It also made me decide how I wanted to do things. So my projects are all about ideas, and it's about making questions. This is why Nature is with a question mark. That project in particular, I'll speak a little bit more about it in a few minutes, is about what do we call nature? Why do we call it nature? And when do you determine if something is natural or not? And because of this, because it's the question that triggers. It's never about the technology that allows me to get there. It's never about the material. Those are just consequences or paths to find the right way to pose the question, the right way to create a trigger for the public to pose questions which belong in the same family. It's not necessarily about asking or wanting the public to ask exactly the same question, but at least to get close to the subject that I'm interested in. And this means that nature was done in an evolutionary biology lab, Tree of Knowledge was done in a tissue culture lab, unbiased used microbiology and plant science. Proteic Portrait was a proteomics project. I have more. Oh, do I? Deacon was about bioremediation and river pollution. The family was using BLAST, which is a software, and also using fluorescent in vitro hybridization, which is a molecular tool. Immortality for 2 was about generating immortal cell lines. Functional Portraits was using functional magnetic imaging. Nuclear Art was again using fluorescent in vitro hybridization to stain chromosomes. Sometimes you can do the most amazing questions with very little technology as well. So growing wheat on a specific page of the Bible can also be about generating a question. But also using very, very contemporary technology like CRISPR, whose developers actually won the Nobel Prize this year. So CRISPR technology is a bit about genetically manipulating life forms, and I have at least 3 projects that are using that technology, 2 of which are done in collaboration with Maria Antonia González Valerio, who's the founder of Arte Mais Ciencia. So to help explain this, I start with empathy. And this is not something you hear a lot in art projects or in stating methodologies in art projects. This is taken from design, actually, but empathize is not a simple thing, okay? What do I mean by empathy? Is empathy the ability to put yourself in another thing's shoes, person, thing, whatever? Or is it about identifying yourself with something or someone else? Is it about just understanding and knowing that a little bit better? There's a lot of things to be said about empathy, and it's not as straightforward as it could be, but I like to think, or I like to state that I start my projects because I like them, because I like to think about the questions that it poses me, and most often than not because it puts me in an uncomfortable position, that I'm not quite sure I know the answer to that riddle. And this is always a fun position for me to be in, at least personally. Then I try to somehow define what is the problem that I'm trying to look at. I try to come up with ideas, and this is where I start to think about what material can I use, what technology do I need to use to make the material and the technology work for the idea that I'm working on. And then experimentation happens until a prototype can be built. And the prototype is the art piece, because the art piece can be rethought. So when I put forward a prototype, it's very important for me to understand how, if the trigger that I created in that prototype is working or not. And sometimes it really, really isn't. And so I need to go back and rethink it, or maybe it gives me another direction for the artwork, and I need to refine that and actually ask a different question with that technology and that material. So this is how I work. So I was telling you, Nature was actually based on the publications in Nature magazine by this group of scientists led by Professor Paul Brakefield. He's retired now. And they were describing how they manipulated the wing pattern of live butterflies. And I was completely overwhelmed how wonderful the technology looked, that they could actually change the wing pattern of the butterflies. Now, it's important to understand, it was a very important thing for me to understand, that the manipulation of these organisms and the wing patterns is not genetic. Okay, so they could actually alter the wing pattern of the butterflies through a non-genetic mean, which means that the manipulated butterflies would have exactly the same DNA as the non-manipulated ones. And this is why the question nature is important here. Because if there is no change in the lifespan of the butterfly, if there's no change in the mating behavior of the butterfly, if there's no scars, if there are no immediate difference between these butterflies and the wild-type butterflies, except because I decided to make them asymmetrical, which would never occur naturally, then the question is, what are the criteria that you are using to determine if something is natural or not? And should we rethink those criteria now with what is being done? I think this is pretty obvious, but still we resist the rethinking of the criteria. And we like to fall into thinking something is not natural just because it doesn't fit a somehow 19th century worldview of what nature is. To show the work, the butterflies are actually very much alive, and you can go into the greenhouse with them. They are in specific conditions because these are tropical butterfly, and they need to be at 27 degrees Celsius permanently. They need to have daylight, night cycles of 12 hours. They need to have an about 90% humidity. So it is pretty uncomfortable for us because 27 degrees with 90% humidity becomes really, really uncomfortable. But it's very interesting. And the plants are there just to keep the butterflies off the floor, because if people go in, they just don't move, okay? If they're on the floor, you have to really watch out because they don't get off the floor. And you can see it becomes a little bit of a performance as well, so I do the alterations to the butterflies at a specific time every day. And what I'm doing under the microscope, you can see on the monitor outside, and you can ask questions while I'm doing it about what I'm doing, why I'm doing, what are the interesting parts of it, how is it possible to do such a thing, things like that. Why are the scientists trying to understand the wing pattern of live butterflies? There's lots of different questions. This project has actually made people tell me things that are absolutely horrible, like you are the devil and you should never be allowed to do anything like this. And other people tell me this is an amazing project, this is absolutely gorgeous and beautiful and all of that. It's really a very emotional project for most people. Now, you saw that I have a lot of projects. I can't talk to you about all of them, or I will if you want to, but maybe on a personal basis. I just wanted to tell you about 2 projects that I did more recently. So one of those encouraging people who told me to write that email to the lab in Leiden is now my husband, and we have been together for about 22 years or a little bit more. And he's an immunologist, okay? He had a medical degree, and then he did a PhD in medical biosciences, and he's an immunologist, but we never worked together until relatively recently. So in 2014 was the first time we worked together. And he's worked with lots of different artists besides me and before me. And this is the first project that we did together. Now, this project didn't start as a collaborative project between me and him. It was actually a project that I was looking at into what does immortality mean? And what does immortality mean when suddenly you find out that an immortal cell line, which is something that is very common in labs and very attractive as a material for research, is actually a cancerous cell line. Okay, so the first immortal cell lines were actually taken from cancer, biopsied from cancer patients. And there's a very famous immortal cell line, which is called the HeLa cells, which was taken from Henrietta Lacks. And there's a whole book, and there's a movie about her cells being the most common immortalized cell line in the world, and also most commonly used in laboratories all over the world. Oops, what happened to the, oh, there. So I was thinking, okay, what does immortal mean? Why did they call immortal cell lines when they're actually cancer cells? And you know, how can we think through this immortality, aging, the wish to become immortal, this desire that we seem to have, and what does it mean to become immortal? And in this case, I decided, okay, can I make my own cells immortal? And technically I could. You need to insert oncogenes into one of your cells and have them grow, and eventually, and you will then have an immortal cell line. There's only one problem with this project, which is for health and safety reasons, and not for ethical reasons, but for health and safety reasons, I am not allowed to manipulate my cells outside of my body, specifically with this technique, but with most techniques anyway. So I was completely barred from developing this project by a health and safety issue, which I'm very, very glad was there, because I don't want to accidentally get cancer, because those cells can be reabsorbed into my body. So what am I going to think about immortality if I can't do this? And then it came to me that, well, why? Immortality is a pretty important thing. If I'm going to achieve immortality in any way, why would I want to do it alone? And this is when I thought, okay, maybe the person I would like to share immortality with, whatever that means and in whatever form we do, would be my partner, the partner I chose for life. And this is when I asked Luis to join me in this project, and he did. But because he's an immunologist, the cells that we had to immortalize were immune cell lines, so cells from our immune system. Now, you may need to understand the immune system is the system in your body that decides if you are you, and anything else that is not you just gets ideally dead. So by making our own cells immortal together or in parallel, we are actually condemning those cells to be isolated for the rest of their lives, whether it's frozen or unfrozen and growing. So we did the protocol together or in parallel, but our cells may never be in the same space together. So for us and for this project, the price of immortality was to be isolated. And the piece is very much about that. So you have two cell flasks in opposite sides of the table. Underneath the flasks, you have a microscope that is feeding live footage to the projectors. The projectors show the cells growing, and the only place where the two cell lines can be together is in the overlap of the projections. Now, we've been called a little bit more radical with this second project, which is Anti-Marta. But in a way, it is very much an homage to some experiments that were done in the 60s by John van Root in Holland, in The Hague. And John van Root is, or was one of the fathers of clinical transplantation. So if it wasn't for these experiments and for the research that they triggered, we wouldn't have clinical transplantation today. Okay, so they were researching rejection, and more importantly, they were researching how to match a donor or an organ to a recipient to minimize that same rejection. And one important factor is actually human leukocyte antigens, which they found had a very important role in rejection. The experiments that they did would probably not have been approved of today, mostly because he just asked some of his colleagues in the lab to exchange skin transplants. And this is one of the images of those transplants. So you have one self-transplant at the end, and then 4 external transplants from colleagues. Next to it, you don't reject your own skin, but you reject everybody else's. Okay, so Luis and I went to a presentation by John van Root in Lisbon at the Gulbenkian Foundation, and after the presentation, we looked at each other and I don't know who came up with the idea first. He will tell you it was me, and I seem to remember it was him, but we just decided not to talk too much about it. We looked at each other and we said, okay, why don't we exchange pieces of skin and make an art project out of it? And he comes up with lots of ideas, okay? Luis does come up with a lot of ideas. Most of the time I tell him, well, I know you're a scientist, but you can also make art, so why don't you make that piece? But this was actually a piece that came very close to my heart. So as I failed to mention before, one of my main interests in terms of questions have to do with identity. I love to think about identity. I think identity is a concept that needs to be rethought constantly, and it's a fundamental concept for making decisions. If I know as much as I can about who I am, what I am, or who I can be, then I can make a better decisions to what I want to be, to who I want to become, and try to get there. So anything that gives me a different perspective, that challenges my understanding of what identity can be understood as, is something that I really find fascinating. I empathize tremendously with. And one of the main concepts in immunology is the concept of self and non-self. And this is a great example of this. Rejection happens because the immune system is there, and if it's a healthy immune system, it will differentiate precisely self from non-self. And so for me as an artist, for me as Marta, the piece was very much about differentiating me from my partner. This is something I need to do periodically because we've been together for 22 years, and sometimes I just feel the need to say, I am me, and forget about this us. Not that we're going to separate, but I just need to reaffirm my identity. It was also very fascinating to me to think that in my understanding with all of this information, the immune system is actually the system in my body, or in any of our bodies, that is more apt to decide what is me and what is not me. And it has nothing to do with my brain. And identity is something that we associate with self-consciousness. But maybe there's a little bit more to it than that. And so this is what we did. So we exchanged pieces of skin. We rejected both. I have the scars to prove it, and I'll have it forever. But This was also a tipping point for me and Luis's working together, because for the first time in his scientific career working with artists, his input towards the artistic concept of the piece was, in my view, more equal to my own, okay? So instead of just me looking into this as, you know, this is an affirmation of self, this is rejection, this is all that, he came to me and he said, well, actually, this is incredibly different from the way I see it. And I know I see it as an immunologist, but actually the understanding of identity and the artistic concept behind a piece is very different from yours. So let me try to explain with him not being here. So as an immunologist, he understands the process of rejection as a very quick step into something else. As an immunologist, rejection happens to create something. It's a creative process. What it creates is antibodies, and antibodies are memory. So actually for him, this is a piece about our love, and this is a piece about our life commitment to each other. Because we did this surgery and we created in each other antibodies that are memories of that person, I will always have within my body a molecule that will always remember Luis, and he will have a molecule in his body that will always remember me. And this is a lifelong commitment, okay? This is not a contract, this is beyond a contract. So this for me, it was really interesting to see how his input in the concept of the piece was so strong. So these are the two works that I've done lately that are ready. I would just like to give you an introduction on a future project that I'm working on. And because it's a future project, I only have the idea behind it. I don't know how I'm going to show it. I don't know what is going to happen. So one of the things that I learned as well in immunology is you have B cells in your body. And most people don't realize that, or maybe I should ask, how many types of cells in your body do you know don't have your DNA? Actually, red blood cells don't have DNA in them. Okay? But the immune system is, like Luis says as well, a very creative system as well. And the immune system creates B cells. And B cells are actually a result of cut and paste of DNA. So the immune system actively creates new DNA for these specific cells. And you have them flowing around in your bloodstream. And yes, most of the DNA that they have is exactly like yours, but there's a part of it that is different and was generated by the immune system. So it actually cuts and pastes specifically. So when I learned this, I thought, okay, in principle, I can take a cell of my body, I can undifferentiate it into a stem cell, and then I can create organs or cell tissues from all of the different organs in my body or cell tissue types, and I would recreate a little bit of a not very functional body outside of my body. Okay, this is actually a technique that is used to study impact of pollution into animals which are endangered, like turtles, okay? Instead of studying the turtles, because you can't just go get the turtles and study the impact, you get the turtles that are already dead or that have recently died, and you then take some cells, undifferentiate them, differentiate them again, and see the impact on those tissues of the pollution. So, but what if I do this with a cell that was generated by my body, so it's mine, but doesn't have just my DNA. It has a part of my DNA, but something that my body decided to make it so, for whatever reason. would the cells and the tissues that I create from that one cell still be me? And how does the identity issue work there? What is the core identity here? So this is a question that I'm willing to pursue. So thank you very much. These are all of the collaborators that I've had along the years and that I'm very, very fortunate to have worked with: scientists, artists, others. I think in general, they're just others. They took their time to work with me, and they embarked on the adventures that I gave them, and I'm very thankful for their collaboration, so thank you too. You have a question?
I have many questions, but I've narrowed it down to two.
Oh.
The first one is the dumb question, which
okay, there's no such thing.
Okay. Antibodies are permanent.
Sorry?
Antibodies are permanent.
Yeah.
Simple as that. Okay. The second question was that I'm really interested in the idea of the individual and where that starts and stops. Where does me end?
Yep.
And whether you thought about that in terms of consciousness or the boundaries of the body or community or those sorts of things.
Yeah, absolutely. You have a lot of questions to be answered by that. It's like, with identity, you have so many ways to go forward. You have, microbiome and all of the biomes. How many? We have less cells that have our DNA in our bodies than we have cells of other organisms. Anyway, so what does me include? And we need to change our understanding of it very, very much so.
I have a question about the B cells. When you have a blood transfusion, presumably there are some B cells knocking about in that blood. Is there evidence as to what
Well, actually, no. So with the blood transfusion, they clean the blood.
Oh, they do. They're cleaning up the B cells.
Of many other things.
But I mean, there must have
Of
been
many other
cases
things.
where they see what happens with the B cell. I mean, it's not exactly the same as the one that carries the DNA that would
Absolutely.
be an antibody. I mean, has there been research to see what happens to it? Does it mutate when it enters another body? Does it get attacked?
I'm imagining yes. I'm imagining yes, because the immune system is incredibly, incredibly precise. But the blood transfusions, okay, so this would be a question for Luis, definitely. But from what I understand, blood transfusions are, I'm not sure exactly what they put. You don't have a lot of cells with DNA in blood. Most of the cells are the hemoglobin cells, and those don't have DNA. This is why the blood is not rejected unless it's the wrong type. And this is why blood transfusions work, and organ transplant does not, just purely. So, but I do think that they purify the blood to have only the items that will not create a rejection. If it does, if there's a cell that has DNA and will generate a rejection, it is probably not a very big issue in terms of body, because if it's just a few cells, the body just eliminates them and that's it. It doesn't even create inflammation or anything like that. That would have to be done outside of a body, but yes.
Maybe even in small amounts with some Trojan horse in it.
There's a project with horse serum that was done by a group in French. They live in Paris. They're a French group. Now I forgot their name, but I can find the reference for that. Where Marion Laval actually had to spend a few months preparing her immune system to the horse blood, because otherwise she would have gone into shock with the injection of horse serum into her bloodstream. And I know this happened over a period of time, and then in the performance she had some stilts that looked like horse legs, and the performance was actually called The Horse in Me. And it was a very complicated process to get her not to go into shock and die during the performance. They had to work with the immunologist to prepare her body for something like that. They can, but it's not advised.
How do they even figure out how to do that? Someone had actually to experiment with that.
Yes. Yes. Thanks. We can do it over dinner.
Thanks so much.
Yeah. Thank you.